Okay, hello 12s. We're talking E2, DNA replication. So, what is DNA replication? Well, it's DNA copying itself, all the DNA copying itself. The process of DNA replication is when you make more cells or when you're growing. So once the egg is fertilized by the sperm, you have fertilization and you have an embryo, and that embryo, the cells in that embryo divide. They divide to make a human being or whatever. That process of cell division, you're going to have the DNA replicating itself every single time. Boom, 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 boom. Replicating. So what happens? What is DNA replication? And why does DNA have to replicate? Well, the why is simple because DNA contains all the information for all the proteins and all the enzymes that carry out the cell's processes, make the cell live. So the daughter cells, if you want to make more cells, they have to contain that information. Otherwise, they're not going to work. One way of looking at life on Earth is basically just carrying DNA for maintaining DNA information. All the things that we do is just so we can have children. Well, our children are just really just our DNA being passed down. It's just DNA. It's DNA going on. We have sequences of DNA that reach all the way back to our bacteria ancestors. It's just that DNA has been passing on and evolving and growing, and here we are. So why does DNA have to replicate? Is to pass on that information so that the daughter cells can function. So what happens in DNA replication? How is DNA copied? So we've talked about the structure of DNA, that it's a double helix with two backbones and complementary bases, A to T, G to C, meeting in the middle with the hydrogen bonds in the middle. And this is the key. This complementary base pairing is the key to replicating, to duplicating DNA. Because what happens, is there a good here? Do we have a good image here of DNA replication that I can show you? Oh, this is okay. Yeah, I'll buy this. So what happens is the hydrogen bonds down the center of that double helix are broken. So by an enzyme called DNA Helicase. So DNA Helicase comes along and basically zips open the DNA. Bzzzt. So now there are these two strands with exposed bases, with these exposed bases. They're not paired. There's an A and a T that are exposed. And then another enzyme, this is the very simple explanation of this. There's a few enzymes involved, but we're only going to be worrying about DNA Helicase, which unzips the DNA, and DNA Polymerase. And what DNA Polymerase does is it comes along and it attaches, there's one DNA polymerase for the one strand, another DNA polymerase for the other strand. And what it does is DNA polymerase grabs nucleotides that are loose DNA nucleotides that are in the nucleus. It grabs them and matches them up. It helps them pair with the exposed strand. And so it helps them, the nucleotides pair actually on their own because it's complementary base pairing, right? You've got, adenine and guanine. They either have 3 hydrogen bonds or 2 hydrogen bonds. So only a thymine can come here and bond with adenine, and only a cytosine can bond with a guanine. But this DNA polymerase helps them into place, and then what it does is it stitches So once one nucleotide bonds here and another one nucleotide bonds here, it stitches those two nucleotides together. So it's making the new strand. And remember, this bond here on the backbone, it's making basically a new backbone of those sugar and phosphates. So it's stitching the sugar and the phosphate of each nucleotide, stitching the phosphates and the sugars of adjacent nucleotides together. That bond is a covalent bond. So that enzyme is making a covalent bond. This process takes energy. And it's called polymerase, right? DNA polymerase, because it's making a polymer of DNA nucleotides. DNA polymerase. So this happens on both. You've got the matching base pairs on both of the unzipped strands, and now you've got two strands of DNA that are the same if DNA polymerase hasn't made mistakes. Sometimes there are mistakes are made. That's called mutations. It's how we evolve. It is the fertile soil of evolution is mutations. But for our purpose of DNA replication, now you have two identical strands. Half of the strand is nucleotides from the parent, from the old strand. It is the old strand. It's half the old strand. And half the nucleotides in the new strand are new nucleotides that have been stitched in by DNA polymerase. Therefore, we call DNA replication or DNA semi-conservative, because an old half is conserved, half of it is old, conserved, and half of it is new. So it's semi-conservative. That's the first step of DNA replication, is replicating the DNA. But I guess DNA replication is cell division. Let's talk about cell division. We're going to talk about cell division. Some issues, some vocabulary. I don't love vocabulary, but let's talk about vocabulary because we need to talk about some vocabulary. Okay. So there's a word in the notes that says chromatin. What is chromatin, you might ask? And chromatin is all the DNA. It's the molecule of DNA, so all the chromosomes in the nucleus of your cell. That is called is chromatin. And it's not just the DNA molecule. DNA is a hybrid. Chromatin is we call it a hybrid. Is that a good word for it? It also contains proteins, and the proteins help organize and stabilize the DNA molecule. And these proteins are called histones. Histones, they're like round proteins, and they help wind the DNA. There's a nice image here. Let's look at this image. I didn't show you the image. Let's look at this image. What can I... Okay, so there's the DNA molecule wound around these histones, chromatin. You can see there's chromatin. It's all the proteins inside the nucleus. Look at all that DNA, all that spaghetti. It makes me want to eat spaghetti. What did I want to show? There it is. So there's the DNA molecule. There's DNA helicase. It splits the DNA molecule. See, it unzips. Those are hydrogen bonds there being broken by DNA helicase. And then DNA polymerases come along and they bring in new nucleotides, new DNA nucleotides, complementary base pair with the old strand, and DNA polymerase comes along and stitches the backbone together. Those phosphate, sugar phosphates get stitched together. And now you have two identical strands. So then what happens? Well, what happens next is amazing. Folks, I think science still hasn't answered all the questions about how all of this works in detail of why and how it happens. Or the details are, I remember studying this, and the details are like gory, boring. Like this factor and this factor. So then what happens? So you've got the DNA has been replicated. Step 2 is all of that replicated DNA now has to be sorted. It's a goop. It's a mess inside the nucleus. So actually the nucleus, the wall of the nucleus that, we learned about the nucleus in the cell, eukaryotic cell unit. So that double membraned nuclear membrane disintegrates. The endoplasmic reticulum disintegrates. All those things around the nucleus disintegrate, and what happens is the new DNA, so each chromosome, some more terms here. So a chromosome refers to all the information. A chromosome is just the information, the DNA information. A chromatid is the physical molecule of DNA and the proteins associated. Those are chromatids. So one chromosome is a chromatid, but the chromosome is just the information. It's the DNA information. So then what happens is, so now we've duplicated all the chromosomes, we've replicated all the chromosomes, and then what happens is all those chromatids, right? So all the chromosomes, so all the information's been duplicated, but all the physical molecules also, with all the proteins and stuff, they have to be sorted. They have to be organized. So what they do is they wind, all the DNA is wound around those histones, wound really tight. It all gets wound really tight. So tight, normally, when we use a microscope to look at a cell, we can't see the DNA inside the cell. It's just such a thin molecule, we can't see all the proteins. But when it's wound during DNA replication, during cell division, when it's wound up, we can see the DNA. Look, I will show you. Here is some, I think these are onion cells undergoing DNA replication. Look, you can see the DNA wound so tight. So normally you can't really see the DNA. See, you can't see anything. That's the nucleus. You can't see anything in there. But here you can. You can see the chromosomes. So they're all doubled up. You can see all the chromosomes. So they get sorted out. The chromosomes get sorted out. And what shape they take, what's happened to them. So here you have one chromatid, which is a chromosome, but the chromatid also is the physical part of it, is the protein and the DNA, and it gets all duplicated. And after it's duplicated, so you have here two sister chromatids. So you have two chromatids for each chromosome. Meow, meow, meow. So all the information is duplicated, and the chromatids are held together by something called a centromere. It's a special piece of DNA that is made that holds them together. This helps them stay organized and not get mixed up, and helps them stay on one side of the cell. This is important. I don't want this coming along when the cell is divided. I don't want chromatids coming along, because now I've got a doubling of the chromosome, and the other half of the cell is missing that chromosome, which is called a chromosomal mutation. It happens. And that other cell probably is not going to work. There's thousands of genes on that chromosome, so that those genes are important. That info is important. And the cell that's got doubled chromosomes, depending on what's on those chromosomes, maybe really have problems, have issues. There is a condition called Down syndrome, where it's a doubling of one of the 21st chromosome. I want to say off the top of my head, I may be wrong. And it leads to some changes, major changes by just doubling the chromosome, because now there's double the information. The gradients, the ratios of proteins and enzymes and chemicals in a developing human is different now, so the human develops differently. Okay, so that's why we have the centromere. It's important because the centromere keeps the two sister chromatids organized. And what happens is the centromere in the cell, when it's dividing, what happens is, here's nice. This is nice, yeah. So look here, all the chromosomes are divided, or they're condensing, and they're all sorted out. The nucleus dissolves, and what happens is these spindle fibers form. These are cytoskeleton, involved with the cytoskeleton. So these huge spindle fibers form, and the centromeres, that thing that's holding the two chromatids together, joins with these spindle fibers, which are just proteins. The spindle fibers are proteins that have secondary structure, but probably not tertiary structure. There are these collections of those proteins. And these spindle fibers help all of these these paired chromatids line up. So one chromatid is on one side, one chromatid is on the other. So that's step 2, is sorting them out. And then, I'm going to include this as a step 3, is what happens is the spindle fibers, the centromeres break, and each chromatid then goes to the other side of the cell. And the cell then can divide. The cytoskeleton organizes this division as well. It's not spindle fibers, but it's just the cytoskeleton pinching off, getting created and pinched off. Organelles inside the cell, so mitochondria, ribosomes, all of those, what are lysosomes, a lot of those organelles and stuff, they get divided up. They don't get divided up equally. It's kind of a messy business. There are mistakes that are made, but the mitochondria come over here, mitochondria come over here. The endomembranous, if you remember from the cell unit, from the eukaryotic cell unit, the endomembranous system, so that's the rough endoplasmic reticulum, the smooth endoplasmic, so the rough endoplasmic reticulum where you make proteins, the smooth, and package them, the smooth endoplasmic reticulum where you make lipid-based molecules and you package them off, and the Golgi body where the final assembly of proteins and packaging happens. They all sort of dissolve and they get reformed in the new cells. With new membranes, they get reformed in the new cells. And so then the cell pinches off, it fixes whatever it needs a fixin'. Maybe there's not enough mitochondria on one side or the other, so it makes more mitochondria and fixes itself, and then those daughter cells chug along. The DNA, all that winding unwinds, it just becomes chromatin in the nucleus, and the cell those cells are ready to go, ready to grow and metabolize, and then divide again. Later on, divide again. Human cells, interesting thing. Some of our cells don't divide. We don't get new cells. Some of our cells last 30 years. Some of the cells in your brain will last 30 years. 15 years for some. So some of you just got new cells. Your skin cells, every 13 days, all your skin cells replicate and are new. The inner lining of your gut is like 3 days. The inner lining of your gut is replaced. Those cells are replaced every 3 days. So there's different rates of cell division. Imagine the Mr. K's brain cells, they lasted 30 years. I'll tell you, the new ones, now that I'm 40, don't work as well as the old ones. I liked my old ones better. Your memory cells, there's memory cells in your brain. They last something like 10 or 16 years, somewhere around there. So your memories I know we still don't understand this. I don't think we understand this very well, of how those memories are passed on. But I'll tell you, as you get older, you make up the memories, or they sort of become these foggy dreams. I tell you guys stories in class. I have no idea if those are real or not. Okay, so is that it for DNA replication? How long? That's a 10-minute video on DNA replication. That's fantastic. DNA, spindle fibers, yeah. So things to keep in mind in DNA replication. 3-step process. So number 1 is you replicate all the DNA. The active members in that are DNA helicase, which zips the DNA open, DNA polymerases, which create the two new strands. The 2 new strands are semi-conservative. Half of the new strand is old, and half of the new strand is new nucleotides. Then is sorting. So the DNA winds itself, it condenses itself around histones, and the 2 chromatids, so the copies, are linked together in the centromere, and then they're lined up by spindle fibers at the center of the cell, and then the centromeres break, and everybody goes to their corner, to their side. And that's the third, that's basically, we're in the third stage, and the organelles are divided up. The cell pinches off. New endoplasmic reticulum are created in both the cells, new Golgi body, and that is essentially DNA replication. It's a 12-minute video. That's so short. Another interesting thing is in like bacteria and viruses, DNA polymerases make more mistakes. They purposefully evolved to make mistakes because bacteria and viruses are under constant evolutionary pressure from the immune systems of the things they're trying to infect, and they make so many bacteria and so many viruses that they can afford to mutate more often because they can have bad mutations, they can have bad errors. It doesn't matter, there's so many of them being made. This increases their chances of getting a good mutation that can defeat the immune system. That's a little extra there for you. That's it.